Hello, everyone, and thank you for joining us for today's BD FACSMatters Ask the Expert webinar on learnings and advancements in high-parameter flow cytometry, presented by Bob Balderas. Bob is the Vice President of Biological Sciences at BD Biosciences. He is a distinguished BD Fellow with over 40 years of leadership experience in both academia and industry. Bob serves on various editorial boards and is also a board member at FOCIS and member of the Dean's Council at UC San Diego School of Biological Sciences. He has published over 25 scientific journal articles and is recognized all over the world as a visionary that helps create academic and industry partnerships for the advancement of science. He has been honored with 2 prestigious BD Howe Awards. Bob also has a passion for sharing best practices in the areas of immunology, high-dimensional biology, and flow cytometry. He has participated in worldwide educational seminars and tours for the last 20-plus years, and we are so excited to have him share his recent learnings and ideas on high-parameter flow cytometry with you today. But before I hand over to Bob, I would like to go over a few housekeeping items. If you have any questions during the webcast, please submit them through the Q&A widget of your console. Our technical application scientists will be answering them on the back end, and after the presentation, we will be having a live Q&A session. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause some issues. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. And if your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help Engagement tool at the bottom of your screen. And lastly, a recording of this broadcast will be made available after the presentation, so look out for an email with additional details on where to access it. And with that, thank you once again for joining us, and I now hand over to Bob.
Well, thank you very much. Good morning, everyone. Good afternoon for those others on the other side of the United States. You know, it's my pleasure having an opportunity to share the next hour of science and technology, and to talk about some of the learnings and the advancements in high-parameter flow. As some of you may know, this is actually the 3rd webinar as it relates to Ask the Expert here at BD. And when they first asked me to do this presentation, I thought to myself, well, maybe we don't always have to think of ourselves as the expert, because the experts are the individuals that are listening to the webinar, especially as it relates to what we're going to talk to today. There's a long history with this, and I'll take a few minutes to kind of preface most of it so that we baseline ourselves with the understanding of where high-dimensional or high-parameter flow, where it came from, and what's happened over the last 4 years. But before we begin here, I'd just like to say that about in 2011, there was a seminal paper, a contribution by Gary Nolan. And Gary, with his team, Aaron Simons and Sean Bindle, some of us from BD, and Scott Tanner from DVS, brought a new technology platform, the Cytometry technology. And it was in a time when we were restricted to about 18 colors in flow cytometry, and we weren't moving higher and higher at that time for a number of reasons. But the Cytometry technology gave us 34 different parameters that we could work with elemental mass spec, and it changed our environment in immunology and in cell biology. And it was a very interesting space, but that technology leaped over flow cytometry from a standpoint of number of parameters. So this is the story of how we all in our industry, in our community as immunologists, came back with flow, because we truly did need a platform that would allow us to not only analyze, but to be able to go back and sort those populations for function. And there's lots of learnings and lots of advancements that have occurred over the last few years. As we start, though, I think what's really important is to, again, kind of paint the picture of where we started. We started with an instrument that allowed us to be able to look at many, many different parameters at one time in flow cytometry, but we were unable to be able to get this to work appropriately until enough dyes were developed. And as that continued, we had hundreds and hundreds of antibodies that needed to be conjugated with all these new colors. And then we needed to find and do work and define the receptor expression for both mouse and for human so that we would know how many receptors or the relative numbers of receptors per cell in many of the different compartments of the immune system. And at that time, there was somewhere around 350, 360 different EDs. We didn't have all that information. It wasn't available in any one space. And then we had to bring all of those things together and create a panel, and that's always been a nemesis in our community of immunology. And it's been something that there are individuals all over the world that are very, very good at it. There's some art and experience, but we didn't have a way and an approach to teach everyone how to do that. And again, we ourselves, as we look at what we do every day, perhaps we needed to learn how to do that too. And then from those CyTOF days, there was data visualization tools that were available from the very first paper where we used SPADE and then TISNE, and multiple data visualization tools became available to us. But it didn't stop there, because there was one central thing that needed to happen, and that was we needed to be able to control our instrument. And whether we had only one in our lab of these high-dimensional boxes that necessitated us to be able to compare data from day to day to day to day, but we also, in some cases, have two instruments in our lab, and we need to be able to match those instruments. In some cases, such as in CROs and in pharma today, they have multiple instruments in multiple sites across multiple countries. And we need to be able to control that too. So all of those things were part of the learnings and things that we had to bring together. And we didn't do it from a company alone. We did it from a co-creation perspective with individuals like yourselves that were feeding back information. And that's part of that story. Well, the other part was the biological side. There are labs that work in mouse models. There are labs that work with non-human primate models. And then there's the majority of individuals today that use these high-parameter boxes, and they're focused in human immunology. Now, when we look at the foundation of those, then there are multiple ways. There are laboratories that work in metabolism. And in the high-dimensional space, the laboratories like John Connolly, Singapore, developed a beautiful array of metabolic markers that are used in flow in high dimensions. There are studies that require us to look at immune mechanisms and also immune function. But as we move through that, then we go back to the diseases, such as autoimmunity and the infectious disease, which to all of us every single day we're reminded of the enormity of the problems associated with different types of viruses. And then there's our cancer problem, for us to look and be able to better understand the tumor microenvironment and where immunity plays a role. But it's not always just those. It's that the pharmaceutical industry and in biopharma, there's a large amount of biomarker discovery that's done in there and in basic immunology too. But we need tools that will allow us to really look deep and lots of different parameters. And then there are those laboratories that use the technology. They use that technology in the in a perspective of high-dimensional flow, or epitope mapping, tetramer technologies. Then we bring all of those together, and we think about the foundational panel optimization. And that's an interesting place for us to start, and one that I wanted to kick off all of the information you're going to see today, because there are laboratories that have been building panels and reporting panels for a number of years. And that is fundamental for all of us. And if there's one thing that probably needs to happen in this high-dimensional space as a learning, is that probably about time for all of us as scientists to work as a community and to share these different types of panels so that we don't have to rebuild them every time we need to ask a question. That we should think about how we build them, the architecture, using backbones and drop-ins to report those in a place. We do have one of those places to go today called the OMIPS. And I'll make some comments about that very, very soon. Now, when we look at all of those things, well, it's in the context of the evolution of flow. And although for everyone that's viewing this right now, it's a BD perspective and the types of boxes that have been, or the analytical boxes that have been available since the mid-70s, there are other companies too that have made significant contributions to analytical flow. And there have been many discoveries and changes that have occurred to get us where we are today, where we're comfortably in a conventional flow, at least from a parameter perspective. We can reach that level of around 50 parameters. But there are other technologies that have come in. That several years back, Sony introduced a different type of technology, a spectral approach to look at flow cytometry. Other programs since then, Biorad and also Cytec, have brought out new instrumentation that challenge us to look and to integrate those technologies into our field. But none of that could have happened unless we had developed enough colors to be able to get to where we are. And it's not just a color, but it's a great color, a color that can that gives us great intensity, such that we can see the separation between a negative event and a positive event. Not just any color for which that stain index perhaps may be very, very small. So there's learnings in here, and we've shared many things about it. And I'd like to take you through that story. So when we look at those 6 different parameters that we need to bring together to actually do an assay, and add one other dimension, which is how we prep that sample, things that we need to think about. That first instrument in high-dimensional flow was a 50-parameter flow cytometer called the FACSymphony from BD. And it was built through a collaboration, a CRADA, with the NIH, in fact. And that CRADA was a unique opportunity to work with academic and government scientists that had great understanding and domain knowledge of flow. And then in the CRADA, it allowed the VRC and Mario Roeder's program to take advantage of early access to that technology to advance the programs that were there at the NIH. The dyes were something else that we needed to really add value to, and there was the acquisition of a company called Sirigen that had built the early brilliant violet dyes with BioLegend. Those dyes then were acquired, the company acquired by BD, and we opened that line up, and we made some of those brilliant violets better, and we introduced the UV line, the 355 line. Since then, we've got a number of new inventions on that side. But then, having all those new colors, well, we had to think about how we could conjugate them. So the biologist and chemist in San Diego worked very hard on a new technology called OptiBuild that allowed us to be able to do fast conjugations to try to expedite the new development or the conjugation so that scientists like yourselves could have access to new colors on all the different specificities, not just BD's specificities, because there's not a single company that has all the right clones and all the right specificities. So we have to use content from different laboratories and different organizations. Well, there was also work in the receptor side, and the work that we did to be able to build some databases. And the very first big database that we built was a program with Primitive Biosciences. That was work with Peter Krutzek and my team. And today it's partially owned by Caprion Biosciences. But it was a way that we took 380 antibodies and looked at the expression of all of those receptors in human blood. Second work that was done by Pablo Engel's team, where so far they've looked at CD1 through CD100. They've looked not only at blood, but other tissues. And then recently, the work that we had done in the mouse was completed with a small company called CEIF, connected to INSERM in Marseille. With the collective work with Bernard Mallison's team and all the brilliant work of Hervé Luche, he gave us an opportunity to be able to look at the expression profiling of mouse with over 300 receptors. Well, the next thing that had to happen, though, is, well, how do we build these panels for this high-dimensional space? And we took the data from receptor expression. We took the data from laser lines and colors and brightness and looked at every instrument we had so that we could use a rational design, a panel design, instead of just the art and the experience of some that are better and more experienced than others. But we wouldn't still be okay unless all those data visualization tools came out. Today there's many. But still, underneath data visualization tools, we need great panel design, reduce spread, be able to see those populations, because without it, those data visualization tools will give us information, but they'll be hampered by improper selection of colors with the appropriate antibody. So the learnings that we had were shared in the early days when the instrument just went out. And it required us to make many changes and rethink what we needed to do to be able to get to able to make panels in a reproducible manner across many laboratories and to share that experience. So today, instead of the experts from BD, what I'd like to do is give you an example from 7 different programs around the world. Some of the early pioneers in this that have allowed us to take a look at some of their contributions, some of the learnings, and I'd like to share them. And within that, then, I think everybody on this line will be able to recognize the fact that there's a lot that needs to go in to doing a high-dimensional panel. More than an instrument, it's more than the colors and the antibodies. It's a collection of many different types of things that have to work and have to work very well. This program that I'm about to embark on here and share with you has work from Mario Roeder, which really, as I said earlier, was one of those pioneers with us that helped us build the instrument. And I'm going to show some of the early OMIT development that Mario and his team did. We'll go through the work of Cindy Ma at the Garvan Institute, Xin Lei's work in Shanghai, Enrico's work in Milan, Pia Kvitzberg's work, and 2 models of infectious disease. One part looking at preclinical SIV models with Marta, and then we're going to finish with some of the real current work that's being done by Rafiq's laboratory at Case Western. That brings us very current to where we are today and faced with COVID-19. As we begin this story, though, I'd like to preface something here, and that is several years back, Mario introduced a concept to us, an opportunity for individuals to build panels and to provide the foundation of what we did and how we built our panels, the biology, the colors, the specificities, and to be able to present that in a way that those panels could be shared with everyone. This was through cytometry. Now, along with that, and the CRADA and the work that we did early on, the most important thing here is the technology helped to answer the questions of the VRC. So a lot of Mario's work was on the characterization of antigen-specific lymphocyte function, in early identification of those protective SIV and HIV envelope antibody responses. And there are many things that program does, but they do work in the development of high parameter and new single-cell measurement technologies. The other individual with him is Thomas, and Thomas is a postdoc with Mario at NIAID, is an individual that took the responsibility to do something very special for our community. And that was, because they're at NIH, there are so many individuals that come from all these different disciplines. So by collectively working with different scientists, they built the specificities that could be important under a variety of different types of headings. And those are from basic immunology, as you see, vaccines of RSV, to TB, to immunotherapies. And each of these panels tell a story. As you can see here, about 5 of them have been published. 1 more has been submitted, and a number of all of them, they're all completed, so it's just a process of moving them for publication. But they cover many different parts of NK T cells, T cell function panels, checkpoint. All of these collectively will be in a format for everybody to look at and evaluate. Because the cost of a panel at 30 plus parameters, well, it's 3 times more than a 10-color panel from an antibody perspective. But from a sample perspective, we can work with smaller samples, much more information out of them. So it's a valuable place for all of us. So I'd like to go through 2 of the panels in Mario's program independently and then show you how they are brought together to show us something very, very important. I'd also like to preface, as you look at some of the early work that I'm about to share with you, that most of the panels from individuals that have contributed in the early papers were broad panels, meaning they allowed us to be able to look at the immune cells from a perspective of T cells and B cells and NKs and monocytes. So it's really kind of the convergence of 4 or 5 panels into one. Now, those panels are not as difficult to build, but the ones that are harder and require more time and thinking about what we do are the deep panels into a lineage. And I'm going to share some of those with everyone too. Now, the first panel I'd like to share with you is the OMIP 51 panel. And this panel focuses on B cells, dendritic cells, and monocytes, and was optimized for cryopreserved PBMCs. And it was that way because the VRC receives samples and collaborative samples from all over the world. And they have to work on protocols to make sure that they preserve those cells in an appropriate way. In addition to the markers that for enabling the analysis, let's say, of the CD14 positive monocytes or the B cell subsets that are characterized by CD10 and CD19 and CD20, 21, and 27. And we add IgD and IgM. and the dendritic cell compartments that allow us to use CD1C, CD11C, CD123, 141, and HLA-DR. What Mario and Thomas did is they included functional markers such as the chemokine receptors. So embedded in this panel are CXCR3 and CXCR5. They also included surface immunoglobulins for IgA, IgD, IgG, and IgM. And then also inserted a co-stimulatory molecule CD40. And then bridged that with the cytokine receptors for IL-21 receptor, BAFF receptor, and TACI. All of which enabled an in-depth characterization of B cells, dendritic cells, and monocytes. Now, using this panel for those cells and defining those subsets and phenotypes in human blood and tissue is a valuable tool to reveal alterations of different subsets associated with infectious diseases as well as in autoimmune diseases. What's also important about this specific panel is that many of the specificities and the clones, I should say the clones, cross-react with rhesus macaques. So those specific clones that were chosen for IgD, CD11C, CD19, CD73, CXCR5, and IgA, they cross-react. So the panel that's here can also be important for the study of immune responses in rhesus macaques as a model of HIV-1. And as we look at the omics, the way that they're laid out, is that the responsibility of the authors is to define the gating structure and to show the biology between which ones. And you can see in the middle of the chart here, when we look at the mature B cells and how they're defined, we can see the other subsets too, the memory B cells, all of which are defined in a very structured way so that anyone could take those specificities. They might need to use different colors based upon the instruments that they have, but the biology and the most important thing is that biological tree has been defined by the gating structure of this specific clone, of this specific panel. And then also other functional markers can be added in there to get and resolve even more information from each one. And as you can see here, even the definition of the PDCs and MDCs, is also done. So this was a really well-defined panel, one of the very first ones that came out. But Mario's team went beyond that. And I'd like to share one more panel on his side because it adds great value to the other panel. So these panels can work together. The OMIP 58 panel that was work done by both Thomas and Mario, this panel enables the in-depth immunophenotyping of the unconventional CD3 T cell populations, such as the invariant natural killer T cells, gamma delta subsets, and the mate cells themselves, CD161 and TCR-B alpha 7.2. And in combination with those subsets of unconventional, we can always bring out the conventional CD3s, defined by CD4 and 8, and the natural killer cells, and defined by the different CDs such as CD16. 6. Now, when we looked at this panel, they also included markers to assess differentiation stages such as CCR7, CD27, 28, CD57, CD45RA, and CD95, and the expression of cytokine receptors such as CCR5 and CXCR5. They added the co-inhibitory molecule antibody for PD-1, as well as activating an inhibitory NK receptor, so by inclusion of CD158, CD244, and CD318 that can be accomplished. Now, as we look at the panel and how it has been laid out, we can highlight several things in this panel. We can see that the mate cells are characterized by that expression of TCR V alpha 7.2 and the high levels of CD161. Now, the remaining subsets of those, the remaining cells of those, after, let's say, exclusion of the mate cells, are the conventional CD4s and 8s, and those cells then can be distinguished and looked at in even deeper. Now, within this same panel, it allows us to also distinguish the 3 NK cell subsets, CD3 negative, HLA, DR negative cells. Now, these are defined, as we said earlier, by CD56 and CD16. CD56 bright CD16s are our early NKs, mature NK cells by positivity for 56 and 16, and then we can read and define the terminal cells by CD56 negative and CD16. And as we move to this next slide, we're able to look at a unique set of markers for T cells, for CD4, CDH, and certain monocyte populations, and we can further investigate the functional characteristics of all of those by inclusion of additional markers that are there. And all of this within a single panel, giving us a tremendous amount of information. But most importantly, what I'd like to point out is that if we look at both of these panels, the OMIT 51 and the OMIT 58, they cover almost the entire cellular composition of human peripheral immune system, all within 2 panels. Get that much information. And if we think about the challenges we have to bring all of those together in small panels and have to compare and contrast them, that's an enormous amount of work for us to do. But the OMIT panels themselves were not just defined to the NIH, and there's many, many labs. In fact, one of the first deep high-dimensional panels was actually prepared by Florian Mayer at the Hutch, where he looked at DC subsets. And that panel has been used by many different laboratories around the world. But I'd like to share with you a little bit more, because it's about the learnings. Now, the very first flow cytometer that went into Australia went into the Garvan Institute. And in that institute, Cindy Ma, who was a group leader in the Human Immune Disorders Program, was one of the very first scientists to partake in that and to begin to learn. And she worked hand in hand with an immunologist in our team by the name of Andrew Lim. And they worked together on a collaborative perspective. Now, most of Cindy's work is really defined and focused on primary immunodeficiency patients. And like the work that was done at the VRC, many of those samples are cryopreserved PBMCs. So it always goes back to how we prepare those samples and how they're frozen and how we thaw them out. Now, one of the trigger points for all the work that was done in Cindy's group is that all of this work exploring all the specificities that were needed to really get kind of a blueprint of really the immune system was that she had about 5 different panels that she had to run for every single sample. By utility of a 28-color panel, they were able to bring them all in to a single panel. Now, Cindy created that 28-color panel, screened for a range of lymphocyte subsets in human PBMCs, primarily, as I said before, in primary immunodeficiency. And this comes with a diverse range of subsets, innate-like lymphocytes, NK and NKT cells, as well as subsets of CD4s and 8s. So she explored the central memory and the effector memory compartments, the Tregs, the TFH cells, the TH1, the TH17. and was able, within this panel, to also add a B-cell compartment to look at the transitional and naive memory cells. Now, as we advance into this slide, it's hard to go through all of this data, and this is data, again, as produced inside an omic, to be able to show the relevance and the gating concepts that go to be able to find all of these subsets. But to me, when I look at this one and I explore each one of those compartments, whether they be mate cells or whether we look in dendritic cell populations here or B cell populations, what's most important for me is that I look at this and I can see the beauty of the orthogonal populations and how she's dealt with compensation across, again, 28 colors. Again, for all of us to understand too, it's a broad panel, and in many ways is not as hard to build as we build with a deep one. But we can look at many compartments functionally of the CD4 compartments, of the naive, the central memory, effector memory, and TEMRAs. All of those can be explored with a series of different types of receptors that, again, provide us unique information about the primary immunodeficiency models that Cindy is studying. But she was one of the very first ones, and it took a while to be able to pull and bring all of that together. So the omics can come from all over, and it doesn't mean that every scientist in the world that uses these is going to publish a paper in this area. But if individuals can take their panels, it's a wonderful, contribution for all of us, because the hard work's been done. And the cost and the time for all of us to reproduce this, to be able to find and build a panel as beautiful as Cindy does, I think if we share this, I think today it is our opportunity to change the way that we look and the way that we share data out there and help other individuals in this space, because it takes time and it takes energy and it takes money to be able to do this work. If we take Cindy's work and we go to the patient itself, here's a great example of STAT3 deficient patients that have reduced TFH and Th17 cells as compared with normals, and you can see within those boxes. Additional to that, on the B cell compartment side, those same STAT3 deficient patients have increased transitional B cells and reduced memory B cells. Again, you can see that as compared with normal donor blood. So these panels tell stories. One of the other things that I've seen across many of these omics is there are backbones. There's backbones of specificities, but sometimes they've chosen other colors. But it's a valuable, exercise in some ways, perhaps, to take all of these omics and begin to see which specificities have been anchored in and build some consistency around those to be able to not have to rebuild all the different colors for all of these all the time. So a little bit of consensus work, I think, could bring great value and great value to all those that are going to use these in the future. As we transition, though, to Enrico's laboratory, Enrico is a principal investigator in the Laboratory of Translational Immunology in Milan. He's head of the flow cytometry core. And there is a linkage here of his accomplishments and his past and experiences, because he was able to spend time at the VRC. And any time that you're able to work in a laboratory where the domain knowledge of flow is just at the top, then really your fundamentals become really, really good. Georgia is one of his graduate students that's there, and they work on the mechanisms of T-cell dysfunction. Now, important here to recognize is that Enrico has used the panels I'm going to share with you in several studies because they explore biology in a different way, and they are not broad panels. Now, the aim they had is to characterize CD4 T cells infiltrating into the lung tumors at the single cell precision. And they need this to be able to identify mechanisms of T cell dysfunction that promote cancer progression. Now, to monitor this system and to use this technology and these approaches with the panels I'd like to share with you, they were able to identify the transcription factor IRF4 as specifically expressed by a subset of the intratumoral CD4 effector Tregs that had high suppressor activity. They showed that this mechanism underlies immunosuppression in the tumor microenvironment. And that is published work that was done just this last year. So the two panels that Enrico's lab uses, one for T-cell memory and the other is a T-cell exhaustion panel. And they're truly deep panels. So this is a challenge to be able to use the principles of antigen density, of brightness, knowing your instrument, the efficiency of every detector that allow us then to pair the appropriate color to the specificity. Now, a lot of Enrico's work is not only in this area, and I'd like to call out the paper that is at the bottom of the slide because it's about the development and the applications and the analysis of high-dimensional antibody panels. So Enrico gave us a present to really learn how he built these, the principles behind that. So these are the tools and the learnings that are shared by scientists that actually help our entire community. If we just look at some of the data that Enrico's team has generated from a tumor, a lung tissue, and blood sample from the same lung cancer patient, and we look at the gating process of the live CD3s and the lymphocyte gates, where those CD4 cells are in the gate, and the Tregs with CD25 and CD127, that's just classical for how we can find those populations. It's quite easy to see that that there are tumor-specific hyperactive Tregs. And by looking at OX40 and PD1 in the tumor, those cells come right out, as compared with what we see in normal lung tissue for the same patient as well as in blood. What's interesting here is it takes all that subsetting, though, to get to that population. So it's much more difficult to be able to find these by much smaller panels. And Enrico's laboratory has also done a lot of work in data analysis and in visualization. As you can see here, the Treg population has been called out in red, and you can just see it stand out there in these different tumors. So those are learnings of going then in examples of deep panels. But there's other laboratories around the world that were early pioneering too, and the first flow cytometer that went into China went into Xinlei's laboratory in Shanghai at the Institute of Immunology and the laboratories of Subin. And they really target and work in Xinlei's lab to look at metabolic diseases. And she too has recently been working on COVID samples, and I thought that perhaps we'd be able to show some of that from her laboratory, but she was still working on that and hadn't completed everything. So she shared with me that maybe it would be best to, from a learning perspective for everybody, to let everybody see what she did first in her lab from an institution perspective, because it's about getting individuals to try things. It's about getting individuals to get comfortable with looking in dimensional space that they've never had before and how to build a panel in their laboratory. So in Xinlei's lab, she shared two different examples here. One of them was just a cell surface panel, very structured panel. And as you see again, the tendency of many laboratories was to move towards these broader panels. And I think it's just an easier way to get into high dimensional space because the panels are easier to build. But because they're a collection of 4 or 5 smaller ones. But you can see by the collective 25 markers that she used, you could find most of the major lineages down to a certain level. So the depth into any of these subsets is not very deep. But it shows us something about all the different lineages, in the immune system in that blood sample. Now, when we look at hers, it was a very structured one. She did not decide to use and build this as an omen. But she was able, as you can see in almost all of these gates, to define cell subtypes very, very well. Her NK or NKT, her monocytes, her DCs, her PDCs and MDCs, all of these defined very, very well and very clean separations. And I think she was exceptionally, proud of what she could do here. And a lot of her work is not only in the human, she's done all of these types of panels in mouse experiments too and published therein. But for her lab, there was a lot of learnings. So being able to move everything into t-SNE, understand what it looks like. And we learned a lot of lessons, because if the underlying flow, as I said before, is not good and there's spread and there's other problems, in how we look at our data to draw our gates and that, well, they contribute to many things that give us different types of perspectives, on our visualization analysis that we do. And also then she used different types of heat mapping to be able to show the expression in different types of samples. That was all on lineage side. What she did on the second panel, though, was to make sure she wanted to be able to use that to explore function of cells. But in order to do that, well, you have to bring together different types of protocols to be able to look at many types of cytokines, or not in this case, but different transcription factors in other ones. But she had to be able to put them all together, but understand the kinetics expression of each one of those too. So she defined a 25 marker panel to look at naive and memory cells, activation and exhaustion together. You can see the cytokines and the specificities and the types of subsets for the NKT and NK subsets too. And this is a challenge to be able to go inside that cell. And this is also an area that we're still behind Cytometry technologies, because all of the lanthanides that could be used can work inside the cell, but not all the colors can, based upon the types of protocols that we use. And with that work, she defined different subpopulations of cells and was able to explore the expression levels of many different cytokines within each of the CD4 compartments and also CD8. So it was time that she had to put into her lab to make sure that individuals could see how to use this box, that it was powerful, and it gave you a chance to look in the cell and on the cell with many of the experiments that people needed to be done in her group. And then again, the same type of process of being able to use and understand the visualization processes to be able to see the underlying work. And to go beyond t-SNE, and you can see her examples that she used here on UMAPs too. So staying current, and it's also something for all of us on the line today. If you're not using these, they help us, and they really pull out populations that can be really important, especially in biomarker work, where we're really looking for something different between 2 or 3, different either individuals or in diseases, where a disease may show something completely different that we can look at and evaluate if patients are refractory or non-refractory to a specific drug. But as we move forward and think about other applications, I said before that there was epitope mapping exercises and different types of technologies. And Cytometry, with the work of Evan Newell, really gave us a great tool to be able to look at this work. But other scientists, when the flow cytometer came out, scientists like Pia, who also was able to spend some time in the laboratory of Mario, where again the domain knowledge in there just amplifies. she has applied this high-dimensional space to look at neoantigen-specific T-cell responses. And she notes, as we all know, that in spite of all the recent major successes in immunotherapy, only a part of the treated patients respond to therapy, and many of those relapse. So she used the high-dimensional flow cytometry to dissect that neoantigen-specific T-cell response. And I always find it very, very interesting in working with Pia is that she's always asking me a question that comes continuously, and it's wonderful, and that is, I need another color. I need a bright color. And what we try to do is push all the new development of every color possible and conjugate it onto streptavidin for her work. And those reagents for everybody as we talk are reagents that may have value in your own laboratories. But Pia has taken advantage of those and asked specific questions in her lab. Are neoantigen-specific T-cell responses detectable in a cancer with a low mutational burden? And are these T-cell responses altered upon therapy, whether it's quantitative or qualitative? She uses that technology to help look at predicted epitopes. And from that, she's able to plot subsets of cells, very small amounts of antigen-specific responses. And as you can see here, she identified neoantigen-specific T-cell responses in 5 of the 7 patients in the specific disease that she was looking. But she embeds all of those different markers. She embeds those into a larger set, structured set for where she can look at subsets exactly, and to go deep into the underlying biology of those to explore that work. So it's interesting going deep into a panel using multiple colors, super bright, to look at very, very small changes in small amounts of populations of cells. Again, she's done just a beautiful job, with that work. As we continue to move through this process and give examples, though, it's important for us to really look at how they are in certain diseases. So we chose to share with you 2 of them that involve viral diseases. And one of them is the work of Marta Catafalmo, who's at the Department of Microbiology and Immunology at Georgetown University. And she does focus all of her work, or most of her work, on the understanding of the immunopathogenesis of HIV. She's able to use SIV models, and she's involved in preclinical studies. And I'd like to share one of those with you, for which attacks were treated with 2 different infusion cycles, each 10 days of IL-15 or anti-PD-L1. And in those preclinical studies to address the potential use of checkpoint inhibitors to restore immune function. Marta's early panel, too, as you can see here, was a broad panel. Again, we see this a lot that's out there. We're seeing more deep panels, but again, the broad panels give us ability to measure and see many different variables in the immune system. She added different function, the interferon gamma, TNF-alpha, IL-2, and CD107A. She followed that up with expression so that she could see PD-1, PD-L1, and L2, AI67. Now, this work that she had done was part of a preclinical study, as I said before, for which there were 2 infusion cycles of IL-15 and PD-L1 in a combination antiviral retroviral therapies. There was a 7-day baseline, a day 49 second for the IL-15, and what she was able to see with this is that both IL-15 and anti-PD-L1, the treatment, leads to expansion of T cells with trafficking and also effector function properties. And the way that she used that panel and then used visualization software to be able to look and identify using FlowSum unique clusters. So the clusters here for the retroviral, the IL-15 as well as the anti-PD-L1 treatments were seen here at day 49 and day 92. Now, they cluster on those 6 different specificities down on the left here with PD-1, with CD107A, interferon gamma, TNF-alpha, IL-2, and CXCR3. These CD8 T cells express CXCR3 and PD-1, that's very low express, and they secrete those cytokines. And they also express the degranulation marker CD107A. So they cluster together. This subset of those cells peaks at day 49 and then begins to go down at day 92 and then at 204 down to baseline. So it gives her an ability to watch what's happening with that therapy. She also did the same analysis on a CD4 population, but the impact was very, very weak, but yet still they were able to see some of the same responses, but to a much lesser amount. And all of this work now has been published by Marta, but it's just a reminder for which you can take many of those specificities, and the challenge is see the cross-reaction of all of those, and then to get all of those into a single panel to be able to evaluate at a fairly high number of specificities in a single tube. I'd like to finish this webinar with one more look at the work of Dr. Rafiq Sakali at Case Western, and the work of Susan Ribeiro and other scientists that are in their laboratory. And Rafiq has been focused on HIV disease, immune pathogenesis, and immunology, immune virology, and immunotherapies for many, many years. And he's been a close friend and a collaborator on many, many big projects. And in fact, during the second cohort in the outbreak of SARS in Canada, when he was at the University of Montreal, we were on a big grant together. And Rafiq was able to move all of the work that we were doing in the different parts of the grant, and we just moved it to SARS to help the community. And once again, it's just another example that individuals, and with Rafiq, the types of work that he does, he's exploring, COVID in a number of different cohorts. And Rafiq was kind enough, although the work is in the process of being submitted or already has been submitted, but he was kind enough to show how some of the panel development that he has is answering questions in COVID today. So he has a couple different cohorts and examples that he'd like to share, and he was happy to share those with everyone. The first one was a 28-color panel. Again, it explored a broad immunophenotyping. It allowed us to be able to look at the different compartments. As you can see, the alpha-beta T cells, the memory T cell compartments, and gamma-delta and NKs, all done in UMAPs. You could see that the SARS-CoV-2 leads to lymphopenia, is very, very open, but the cohort that he was working with when he evaluated all this was the work that he has done with mothers and cord blood. And they were able to do immune subsetting in these fresh bloods from delivery time point for both maternal and cord blood, and it was assessed in a way and analyzed with that panel. You can see by the circles that are here and the density plots that there's distribution of different populations in different places. And if we look at the histogram of the cluster counts in both samples, we can see that they too can be compared. Most important for us here, it shows that there's significant differences in here, defined subpopulations in both maternal blood and in the cord blood of those individuals. And by taking all of those specificities, one can look at important data sets in the modulation of the immune cell populations and between both cord blood and mother. And it's very, very clear that in cord blood, the V-alpha 24, V-beta 11 natural killer cells display, a Th2 type cytokine receptor profile. All of this data is coming out of that cohort and that panel that Rafiq's team had developed. But they also look deep using a T-cell exhaustion panel. And within that specific panel and the evaluation of a different cohort, they were able to look at the characterization of high tox and low interferon gamma during exhaustion. And they profiled that out using a variety of different types of cytokine readouts, receptor readouts, to show effector differentiation over time. And all of this is valuable information that we could never see unless we were able to bring all of it together to see how it works in concert. So when we take collectively all of those different examples from investigators, the experts in the world that have shared and worked hard and been able to be successful, it wasn't just about an instrument. And it was really about a collection of all that work with dyes and antibodies and understanding of receptor expressions, and then trying to build panels and getting all the antibodies in that can give rise to looking at many, many different types of either disease processes or metabolic processes, or, as I showed, for epitope mapping, and at the very end of the day, how some scientists take their panels and they build other papers for us so that we can read and use those panels. So to conclude today, I'd like to really highlight the contribution of many individuals. And this program is almost 4 years old today, and there's hundreds of boxes in the community all over the world. Some of these individuals that are listed here were part of the original team that Larry Duckett had with David Matsuyama and David Doe and Austin Wu, the engineers that worked together to actually build the original symphony, and how we went out and tried to demonstrate the utility of that instrument to many of the scientists that I showed their data today. There's other individuals in here under Bill Peck's team that have done just a fantastic job in the United States for laying out the foundation of how to use those instruments, and also in Europe with Jens Flescher and all of the application individuals that have helped and worked with all the different teams all over that you saw today in China and also in Italy, and the team of Andrew Lim that's worked with Cindy Ma over time. I really want to say, thank you. And I know each one of those investigators has appreciated the impact and help that you've given them. But nothing's better than to say thank you to Mario and Thomas, Cindy Mott, the Garvin, Enrico in Georgia, Pia, Shin Lee, that we have lots of different discussions about how to apply this technology, and looking forward to the new publication that's working on and just about to go into submission. Marta, always the conversations around the work in SIV and the work that has to be done when you don't have all the antibodies you'd like to have, and really for the years of contribution and time and grant writing and sharing and experiments with Rafiq Sakali. I'd like to thank everybody and open this up for questions. Thank you very much.
Thank you, Bob, for that fantastic and insightful presentation. We are now in the Q&A portion of the program. However, on your screens, you should now be seeing a poll question. We would really appreciate your responses. And if you are unable to view the poll, please refresh your browser, and it should show up after that. So joining us for a Q&A session are a few additional experts. First, we have Nathan Wizen. He is the High Parameter Scientific Solutions Manager at BD, and he has been with BD for the past 7 years, moving from a flow cytometry instructor into his current role. And we also have with us Ravi Hingarani, Senior Scientific and Application Support here at BD, and he's been focused on market development in immunology at BD for the past 22 years. So welcome to you both. Okay, so I'll get right into the questions. And our first question is asking, how can we define gating and assay controls for high parameter flow panels that can be implemented in a QC environment, particularly for markers and samples that do not have clear separation of expression? So Bob.
Okay, I'll take that. First of all, thank you everyone for staying with us during the last hour. I think that this question is a very important one, and it deals with, there's many questions in it actually. When we control our experiments at 8 and 9 and 10, 11 and 12 colors, there's a certain process that we all do and we all use. But here the real problem occurs is that when we get to 25 and 30 and 35 and 40 different specificities and colors, the enormity of how to control those experiments, oftentimes, is very, very challenging. And lots of the opportunities we have with high dimensional space have everything to do with the fact that we can get a tremendous amount of information out of a very small sample. So you have the two dimensions, right? You have lots of opportunity to get a lot of information, but how do you control that? So we work with many laboratories all around the world and address that in a variety of different ways. But one of the interesting ways about approaching that is that we look at the biology and you can control specific subsets or specific specificities by what we use as a marker -1 approach. And this was a very interesting way of looking at it, and it was done by Aaron Tisnek, and I appreciated it years ago when we laid down a formation for utilizing multiple specificities on a unique color across different lineages in the sentinel type flow approaches. And again, we were in a position where we had lots of controls we needed to do, but it's very, very difficult to do so many of them. So by picking specific ones through the range of, let's call it antigen density, from high expressors to lower expressors, or in specific specificities that maybe there's a lot of diversity of expression, you can control those by adding marker -1. And if anybody would like to see how that's done, you certainly send me, an email to robert_balderas@bd.com, and I will address that for you and show you how to set up those types of controlled experiments.
Thank you so much, Bob. Nate or Ravi, do you want to add anything before I move to another question?
Yeah, hi. So one other thing I just wanted to add is if you do have any other questions or anyone has any other questions about how to set up some experiments, what kind of controls they might need, no matter the experiment or the instrument you're using, you can always contact our research applications group. They're a group of scientists who are happy to help and can help you with any needs that you might have.
Great. Thank you for that, Nate. Okay, so another question is around some of the examples that you showed in the presentation, Bob. So Amit Singh is asking, these examples of UMAP or TISNI are representing one sample or many samples. And they want to know what is the best way to represent data of multiple samples. And he says, for example, I have 10 normal and 20 patient samples with a 22 color panel.
Okay, again, another really good, question, and it certainly is a challenge for us. But the approaches of using, TISNI and UMAPs are exceptional, approaches to be able to visualize our data. And The one thing that you see about high dimensional space, different than where we were with 18 colors, is that as you add more and more specificities, and this is in a deep panel, many of these specificities are very, very low expressed. And what happens is the diversity of expression down at 25 and 30 and 35 or 40 colors in that range, many different receptors are either so low expressed or completely gone. So there is great advantage in concatenating multiple samples together and then to separate that data on a per sample basis to make sure that we can visualize and see where specific subsets of cells are. So I think there is always advantage of doing your work, concatenating, downsampling to a certain level. And again, our FlowJo team, our application teams in San Diego and throughout all over the United States and the world does a really good job with this. But it is something that I would say there is great advantage by bringing all the data together into a single sample and then breaking that data down on a per sample basis, because the diversity of expression is so wide. And I think this is actually a real good question as it relates to asking the question, what is a normal, and how many normals do we need to often put into our controls? I've shared some data that our team in Australia addressed by looking with 15 different samples. This was done by Andrew Lim across multiple instruments, and we can see that there are complete deletions of unique specificities. And when we see that in there, I think, and then control an experiment with one or two controls versus, let's say, a cohort of individuals that may have a disease or whatever the question is, I think that much more time in high dimensional space has to go to defining normal populations before we may be even able to accurately look at disease samples. So I think the question's out there. How many normals do we need to test in high dimensional space? I don't have that answer yet, but I don't think it's one or two, and I think it's many, many more.
That's great. Ravi, Nate, do you want to add on to that? Okay, and so our next question is asking, where can I find this database on antigen density of mouse CD?
Okay. First I'd like to thank Hervé Luche for being a fantastic collaborator in the laboratories at CIF under the leadership of Bernard and Marie Mallison. And it's just been a great couple years. And also I'd like to thank my BD European office and for supporting the initiative and making it possible for Hervé to do this work. We have now finished everything. Just recently, Hervé had done a tremendous amount of data analysis, and we're just looking at all of the data in completeness right now. As you would expect, there's a specific amount of QC that we have to do within the database. We will be publishing this work very, very shortly. But I think if there's anyone that needs specific data, you can address an email to me or to Hervé, and I will make sure that if I know who had asked this question or if others would like to know it, I'll make sure that both our emails are on there. The data is actually quite rich and is differently done than we did in the human. It's done with a single mouse strain with a greater than 1 N number for all of them. It's done with an anti-CD3 stem, so data was acquired, and we actually, Hervé's group also injected the mice with PD-1 and looked for an inflammatory response. So the diversity of the data is really, really big. It explores multiple factions of the immune system as it relates to inflammation. So we can see many markers that no one knew were either up or down regulated in these types of responses. And again, we were able to quantitate that that information using the standard, quantum bright technologies with and without the use of neutral density filters. So there's a little interesting twist that was in that data. But that data will soon be available. We have to get together as a company, as two different companies, and my European office to make sure that it gets formalized and done in the right way. But we will be having that information available for others to be able to see.
Oh, I'm sorry. So another question for you, Bob, is from Jason Killo in Merck. He's asking, do you find many people taking advantage of sentinel gating in high parameter flow?
So, I would say yes. I think that there's more and more of it that's occurring all the time. And I think where the sentinel approaches really help us is it helps us when we're using broad panels with multiple lineages. So anytime you're on a T cell or on a B cell, let's say, and if we know by looking in these databases whether or not a specific protein is either expressed or not expressed on different lineages, we can use these approaches inside. And what that will allow, if we just think about it for a minute, if we had a 25 color experiment and we had 5 lineages, well, that might mean that we could only go down 5 different specificities per lineage. But if we use sentinel gating approaches within that, you could easily get your 25 specificities up to 40 specificities just by understanding where those specificities are expressed. But what is always important is to realize that working with a normal human or a mouse, everything seems to be okay. When we're working in cancer fields, then there can be specificities that are expressed that we might not understand that or might not see it in normal biology. So one needs to always be careful with it. But I'm seeing much more of that being used in panels today. Recently, there was a publication for which the work, I believe, was done in Honest Larvae's laboratory at SINE. So there are a number of them that are starting to come out, but I see lots of that practice inside panels today, but always in broad panels.
Okay, good. I want to allow opportunity for the other experts if they have anything else to add to that response or that question. Okay, and so another question from James Simone at the NIH is, what are the prospects for expanding the fluorochrome color palette beyond 30% to be? Beyond 30%, I think is what they mean.
Beyond 30 colors as they are right now today?
Yes.
Yeah. So I think that there is a lot of hope. There, anytime we build colors, there are different things that we are challenged with. Number one is not always about a new color with a new spectrum. It can be correcting old colors. It can be moving colors from one laser line, as we have been looking at, and moving colors off of, let's say, a blue line and moving them onto a yellow-green line. So some of those new ones are in the process, could be coming out. They're also opening up colors off additional laser lines, and we had done some work and experimented off of that deep UV, that 300 to 320 line. So it's applying the technologies to there. But I think that the real interesting question here is how the field in the future goes ahead. And I don't mean just moving from where we are today with conventional flow cytometers and just everybody moving over to spectral. But the spectral is a very interesting technology base. and it allows us to be able to take advantage of colors that have similar spectrums. So when I look at the technologies that we have, let's say with Sirigen today, in the conventional space, we take time to position them between each other. And we know that because that's the way we've done and used filtering for many, many years in flow cytometry. I think as we look at the spectral windows out there and the technologies that allow us to actually work, we're going to see that we could build many more really good colors utilizing the technologies of Sirigen that we've used over the last 5-6 years. And additionally, some of the new scaffolding technologies that are starting to come out too. All of those technologies are going to expand. But I think for everyone, we have to remember it's pushing those colors to a certain level does challenge us in how many. And it challenges also as manufacturers to get the colors on the specificities and to make sure they're quality controlled. And it goes back even to addressing how we standardize those laser lines with the right types of beads for every color if needed. So I think there are as many, many more things we think about. I think the answer is there will be more colors. But I think of, when I look at our field in immunology, though, I think of deep, high dimensional biology. Me today, I think the work that was done by the New York Genome Program in the Site-seq work, the antibody-seq technologies, the total-seq technologies that are out there today, by using and taking advantage of an antibody conjugated onto an oligo, that there will be a teetering point at a certain point, whether we need more colors or we use a sorting upfront technology connected onto looking at multiple specificities, because I think we've proven today that we can look at excess of 100 specificities by adding antibodies to oligos. And at that point, we're not working with colors. We're not working with spillover at all, or spread, or any, anything in that side. We're looking at sequence data. And not only is that sequence data, very interesting, but it is quantifiable and can go back to the numbers of receptors. But it's the scaling of our field in immunology that it could be that the color side is going to reach a certain number where other technologies can append onto the other side and just bring us great wealth and information. But it is dependent upon lots of experimentation, learning how to use it, the expense and cost of sequencing that's all out there. But if there's one thing that we all know, and that is that field of sequencing costs have gone down and down and down, and I would expect it to continue over time as new technologies come in. So maybe a long answer to a very interesting question, but I would say there will be new colors. They will all come out, and they'll be used by many people. But it's also important to realize that you need to make bright colors, and you need to make them really, really well, and you have to have them available to be conjugated onto many, many antibodies.
That's really great, Bob. I think as we're thinking about, the future and how do we advance, another great question from Ilaria Russo at Keele University is, how can we advance from being limited to take snapshots of the population to visualizing cells over time in order to see their temporal changes?
I think one of the easiest ways today, I think, is if I go back a few years to a lot of the work of Evan Newell, and that he did, and he used at these, in the Cytoph technologies. And I think that the same basic, approaches have to be done, whether it's on Cytoph or whether it's conventional or spectral flow. I think the whole secret of what we're going to be able to see today is in those tools of visualization, that you can track them. Just the wonderful tools, that we could see out there, and I can give a few. It's the work of gemstone, right? To see those temporal changes over time. There's many approaches, not many, but there are a number of approaches today that give us the insight in there, but it's learning how to use those tools together. And I think the whole field of bioinformatics and individuals that just are showing us how to do all of that work. I think we need to get closer to the individuals that can really spin in the data world for us. I think we do have the tools today as it relates to the technologies in that CyTOF space or spectral or conventional flows today. But I think our weaknesses as a community of immunologists is we haven't grasped enough of those tools to be able to see those temporal movements and everything in flow cytometry.
Great. So I think we are at the end now of our Q&A session. So thank you, Bob, and all our experts, Ravi and Nate, for being with us today. Thank you, audience, for all the great questions. We, if there was a question that we did not answer, our experts will email you an answer if we didn't get to it in this session. And as we wrap up, please note that CE credits will be given to attendees, so look for an email with more details on that. And also a recording of this presentation will be available on the webinars playlist of the BD Biosciences YouTube channel and also on the webinars page of bdbiosciences.com. And with that, thank you so much for joining us. Thank you, Bob, for a wonderful presentation. And we hope you found this insightful, and we look forward to seeing you on another webinar. Have a great day.